Introduce you Bert Horst. He is working as a professor at the Erasmus University in Rotterdam, and he just told me it's like, well, kind of different ways. He came out to the biological clock and is working on genetics, and is now working together with many groups about how our biological clock works and about the influences on our lives and aging, etc. Well, I don't tell because you're going to tell us. Thank you, Bert.
Thank you, Ella, for the introduction. I would like to thank you and Jonna for giving me the opportunity to present my work on this meeting. So when I first got this phone call from Jonna, I thought, okay, how on earth can I be of help to this master course that you are organizing? But then Jonna was explaining that this was a mixture of disciplines, people come from different disciplines that meet in this session and try to see how they can collaborate. Then I explained a bit about my work, and then she said, okay, this is beautiful, this is exactly what we would like to hear, interdisciplinary work. So I hope you will appreciate my presentation for tonight. And hopefully it may also give you ideas to work with in this creative lab, let's say, session. So what I would like to tell you about is the title is the circadian clock, and I'd like to start with a small movie. This one. Okay. So this movie showed you that apparently living organisms do have something with time, maybe even have an internal clock. And that this is the case is also very evident from this slide over here, our sense of time. And it shows you that when you make a transatlantic flight and you, for instance, fly from Amsterdam to New York, at your destination, you kind of feel out of place or out of time with your environment. So you want to sleep when you have to be awake, and you can't stay awake when you have to be active. So we all know this as a jet lag, and this again suggests that we do have, maybe you have kind of internal clockwork that kind of generates time. And that this is the case was first shown for human beings by Jürgen Aschoff, as is shown on this slide. So Jürgen Aschoff had access to what we call time-free bunker. So it was a time and sound proof environment where he could house volunteers for, let's say, 4 or 5 weeks. Those volunteers were allowed to live according to their own, let's say, feeling of time. So they could switch on the lights whenever they wanted, they could go to bed whenever they wanted. The only thing they were deprived of was time information. Now, when Jürgen did this experiment, he could show that if you allow a person, let's say, to follow its own, let's say, internal rhythm, and as you can see here, he recorded the sleep-wake cycle. You could see that if those people are allowed to kind of live the day the way they want to live it, you see that every day the activity starts 1 hour later. You can calculate from this line that apparently this internal cycle of activity and inactivity, the sleep-wake cycle, has a period of about 25 hours. And this is what we call a circadian cycle. Circadian, where circa comes from about, and 1 day dies. So it's an internal rhythm with a periodicity of approximately 24 hours. Circadian clock. We call it circadian clock rather than biological clock because we do have many biological clocks in our body. For instance, if you look at females, you have, for instance, the menstrual cycle of 28 days. But also when you look at your heartbeat rate, your you name it, the frequency at which neurons fire, your breathing frequency. These are all driven by kind of biological clock. But the clock that refers to the day-night cycles, the day-night rhythms, is known as the circadian clock. Now, the sleep-wake cycle was one example of a rhythm that was driven by apparently a kind of internal clock. As you can see from this slide, there are many more parameters that are under control of apparently the circadian clock. And for instance, right now you may have your highest body temperature. 1 hour from now, your melatonin secretion will start to take place, or you may kind of start to feel a bit sleepy. And so you can find many examples of parameters in behavior, physiology, and also metabolism that are controlled by this clock. For instance, the body temperature, high and low, high during the day, low during the night. Renal function, high during the day and low during the night. You name it, it's under circadian control. What could be the purpose of having a kind of internal timer? That is kind of illustrated by this slide over here. So you can have a student apartment or your house, whatever your flat. It may have a central heating system, and you may have this kind of classical thermostat. So what happens? You wake up to your cold house. You first have to get out of bed, switch up the heater or the thermostat, and then your house starts to get warm. But wouldn't it be way more comfortable if you would have an electronic thermostat, which means that you can allow your house to warm up before you actually wake up. So this gives you an advantage. So it helps, in this case the house, but in the case of a human being or another organism, it helps the organism to anticipate on the special physiological needs it will have during specific moments of the day. And for instance, before we wake up, our body temperature already raises. So this is good for us, good for living beings. And apparently nature finds this that important that it has kind of conserved these circadian clocks in evolution. So whether you look at simple molds or algae, frogs, so amphibians, fish, but even plants, flies, mice, humans, we all have a circadian clock. This circadian clock, so being prepared on the things of the day that are coming, help us to survive. Now, here you see our favorite subject that we study in Rotterdam, that's the mouse. You can see here that also the mouse has a circadian clock, and what you see here is a so-called ectogram, which means that we have plotted the activity of this mouse in a running wheel on day 1, day 2, 2, 3, 4, and so on. So this is a double plot of running wheel behavior. And I hope you can see it on this slide, it's not that clear, but right here you see the day, the night, day, night, day, night. What you can see is that under a regular light-dark cycle, this mouse is active during the night, it's a nocturnal animal, and inactive during the day. And if you now, from here on, leave the lights off, you start to see that this mouse is continuing to be active and inactive with a rhythm of approximately 24 hours. You can calculate from the slope of this line that this mouse has a circadian periodicity of 23.5 hours. So it's also showing a circadian clock. But now we are faced with a problem, and it is that our day-night cycle on Earth is exactly 24 hours. While these endogenous clocks apparently have a periodicity of about 24 hours, 23.5 hours for this mouse, 25 hours for human beings. So if nothing would happen, we would continuously live in and out of phase with the light-dark cycle on Earth, which of course is very unhandy. And then I always say nature wouldn't be nature if it wouldn't have found a solution for it by itself. And the solution in this case is light. As you can see here, it's literally light. So it's a flashbulb for the here, it's literally light. As you can see here, what we know now is that mice, but also human beings, they receive light information via photoreceptors in the eye. So we have our rods and cones, and in addition to that, other photoreceptors in the eye that can receive light information. They can transfer this light information to the brain, and then this light information is used by the circadian clock to reset this clock every day to a light-dark cycle. As you can see, this is the reason why under a light-dark cycle, this mouse is nicely behaving according to a 24-hour rhythm. So whether you have a short, or let's say a fast or a slow ticking clock, light every day resets this clock to stay in phase with the light-dark cycle. And then you can ask yourself the question, okay, apparently we do have a clock, where is this clock present? And as you can see from this slide, so this is a mouse brain, you can make sections through the mouse brain, and then you see on this section an area that we call the suprachiasmatic nuclei. It's an area of the brain that is located just above the optic chiasm, so that's the place where the two optic nerves cross each other, and then just on top of that position in the brain, in the hypothalamus, you see this area of, let's say, 20,000 to 25,000 nerve cells that apparently produce time. And that this is the case was shown in the early 70s by Moore and Eichler and Stefan and Zucker. So what they did, they took a mouse or a rat, they inactivated this area by inserting a very tiny needle into the brain. With a stereotactic microscope, they could guide this needle to specific areas in the brain. Then, by giving a small electric pulse, they could kill the nerve cells that were at the tip of this needle. And when you destroy this area, we know it's superchiasmatic nuclei, you see that you get a mouse that all of a sudden doesn't show the rhythm anymore. Yeah, I never would have done this experiment myself, and you for sure cannot do this experiment with human beings. As you can see, humans also do have an SCN, so you see this area over here in the hypothalamus. You can make a cross-section through the human brain as well, and then you can see here this area of about 25,000 nerve cells that produce time. So humans also have a superchiasmatic nucleus.
Same amount as the mouse.
Sorry?
Same amount as the mouse.
Yeah, it could be the same amount or slightly more. I mean, but it's let's say it's not a huge amount of cells. It is around, let's say, 20,000 neurons. I forgot to mention it. If you have questions, so please ask them straight away. Better to ask them when I'm presenting and showing things than leaving them till the end. So.
Is the clock evolutionary, or is it caused by having a day and night rhythm? Like with a mouse, it's 23 and a half hours. But if a mouse never sees daylight, does he develop the same rhythm?
Yeah, that is true. So every organism, virtually every organism on Earth has a clock. Even organisms that are, let's say, living in let's say, the deep ocean areas, and there is a reason for that, and I will show you that in a moment. But the idea is that even those animals, it has been shown that even those animals do have a circadian clock. The only thing is that those deep sea organisms, they don't see, literally they don't see the light, so their clocks are not synchronized to the day-night cycle. But still, it's advantageous for those organisms to have a circadian clock, and I'll come back on that in a moment. So we do have a clock. Then, of course, the question is, how does this clock work? So, how can our body generate time? And to answer that question, we should look like to introduce you slightly into, let's say, some molecular biological techniques and aspects. I don't know how many of you have received an education in biology, but what you see here is a cell with a nucleus. In this nucleus, we have our DNA packed in chromosomes, and this DNA, this, let's say, coat of, let's say, this piece of this huge molecules, they are composed of, call it this nucleotide ctDNA, and those four letters can generate the genetic code that allows, let's say, the synthesis of proteins. So we have some 25,000 genes, and this genetic information can be used to produce these various proteins. Those proteins by themselves can form protein complexes again. So how can our body, by having this DNA and by making these proteins, how can our body generate time? So this might be a bit complex, but I hope you get the point in the end, and in a moment we will get back to more simple things again. So here is where genetics can be very much of use. So basically, if you want to know which genes are involved in time generation, which proteins are involved in time generation, you should be able to first identify those particular genes. That's why genetics comes at help. Genetics makes use of organisms, and whether these are mice or yeast or plants, in which certain genes are defective. They don't work anymore. And this can be due to spontaneous mutations in the DNA, or it can be deliberately made by, you can, in the lab you can also inactivate particular genes, for instance, in a mouse or in a yeast. So if you have an organism with a certain gene inactivated, you can look at how such an animal would behave under specific tests. So basically what we were interested in, how do animals with specific gene defects perform in, for instance, this wheel running setup. So are there genes that when inactivated give a disturbed circadian clock? Now we were talking about interdisciplinary collaboration, we were talking about thinking out of the box. Now this is, I think, a nice example that shows that if you keep your eyes wide open and if you're open to new ideas and new visions, yeah, you can sometimes have very nice results. And so, as you may have read in the introduction in the folder, so I was originally working on mouse models for genes that were involved in the DNA repair process. So, for instance, when we walk outside in the sun, our skin is continuously damaged by UV light. It causes cross-links between two adjacent nucleotides in the DNA, particularly those C's and T's. And what you can see here is a piece of damaged DNA. So there's a cross-link between the nucleotides. Now, these cross-links are very dangerous for a cell or for an organism, so that's why evolution also has generated processes or mechanisms to repair this DNA. So what we are looking after here is a process called photoreactivation. You see here a DNA lesion that is introduced by ultraviolet light. And what happens here is that the protein, a photolyase, can bind to this DNA damage. And then this photolyase molecule contains two called core molecules. One is serving as a kind of antenna that can harvest light energy. Then this molecule is transferring this light energy to another molecule in this protein that uses this energy to split this bond between the two pyrimidines. So what happens is the photolyase binds to the DNA, then with the aid of visible light, it repairs the DNA and then it dissociates again from the DNA. So it's a repair enzyme that is present throughout nature. It is evolutionary highly conserved. There's only one but, and it is that kangaroos still do have those repair enzymes, but placental mammals like men, mice, so we have lost this repair enzyme. Okay, now given the fact that during let's say the splitting up between placental and non-placental mammals is a pretty young event in the evolution, we thought, okay, we should have a closer look at the DNA of humans and mice, because those photolyase genes should still be present in our DNA. So we did a search of databases and we did some cloning experiments, and indeed we could identify two genes in men and in mice that resemble these photolyase genes. So this is a cartoon of a photolyase containing a 500 amino acid core backbone with an extension at the start of this protein and the N-terminal site. However, when you now look at those two genes and proteins that are encoded by the two genes that we have isolated, you see that this core structure of this protein is still the same, but instead of having an extension at the front, they have an extension at their back. So those proteins are the same, but again, they're also not the same because they differ at the extensions. And now the interesting thing is that, in fact, those photolyases form a protein family of very similar proteins with the cryptochromes. And cryptochromes, by the time we found those proteins, those genes, were only known to occur in plants. And in plants, those cryptochromes act as photoreceptors. So they take care that, or they are responsible for the fact that plants only grow in the light period. They're also responsible for the opening and closing of the flowers. So in plants, those proteins are photoreceptors. But now how about mammals? So since those plant cryptochromes are photoreceptors, the first thing, of course, to think of is that those proteins in mammals, in men and mice, should also work as photoreceptor proteins, so they should be able to catch light. However, if you think about where we would need photoreceptors for, of course it's vision. You can see me, I can see you. That is because we have opsins in our rods and cones in our retina in the eye. So vision is not a very likely function of the mammalian cryptochromes. And I forgot to mention that you can see that we found two homologs of these photolyses. Try one and try two, cryptochrome 1, cryptochrome 2. So what would be the function? Now one of the things you have to think of, so here you have really to think out of the box. So one of the things that we thought when we first saw those proteins, that, okay, where do we need photoreceptors? So by the time we found those genes, nothing was known about how our circadian clock is reset by light. So then we thought, okay, maybe those proteins are not involved in vision, but maybe these are the photoreceptors that we need to reset our clock. And I still remember the moment when I proposed this idea in our work discussion at the lab. So my boss, Jan Hoemakers, was laughing, oh, you can do the experiment, but I don't believe in it. So anyway, so I proposed, okay, maybe these are the photoreceptors, so let's put the knockout mice that we have, because we in the meantime also inactivated the CRY1 gene and the CRY2 gene in the mouse. So we started to do circadian experiments. We placed these animals in a setup with a running wheel, so we could monitor the activity of those animals. Here you can see a normal mouse. I hope you can see that the onset of activity in this double histogram slightly moves to the left every day. So you can calculate from the slope of this line that this mouse has a periodicity, clock has a periodicity of 23 hours and 45 minutes. How about our CRY1 knockout mice? That was the next experiment. As you can see here, we noticed that surprisingly those animals had a very fast ticking clock. So when you look at the periodicity of this circadian clock of this CRY1 knockout mouse, its periodicity is 22.5 hours, so more than 1 hour shorter than that of its, let's say, healthy wild-type litter mate. Now we not only had the CRY1 knockout mice, but also the CRY2 knockout mice. So yeah, how about the CRY2 knockouts? Completely the opposite. So those animals had a slower clock. So this already suggests that the CRY1 and the CRY2 proteins are kind of interacting and giving, in fact, kind of a period to the circadian clock. So they're involved in the generation of, let's say, period determination. Now then, of course, the advantage of genetics is that you now have CRY1 and CRY2 knockout mice. You can put them in the same case, male and female. They mate, and after a couple of breedings, you can generate mice that don't have CRY1 and that don't have CRY2. So they're completely deficient in CRY1 and CRY2. And that was even more surprising, because as you can see from this one, those animals don't have a rhythm at all anymore. So they were completely arrhythmic in constant dying. So when those animals were allowed to follow their own clock, they didn't have a clock. They were arrhythmic. They were sleeping and active at, let's say, random times. Okay, then you all say, how about the top part of the figure? It looks pretty normal over here. Yeah. Right now we know that animals, mice, they are animals, so they show what we call adversive behavior when the light is on. So if the light is on, they don't want to use their running wheel. So it seems as if they don't have a circadian, and they still have a circadian clock over here. But would you replace a running wheel by another device to monitor activity, you would see that also in the light-dark cycle, when the light is on, those animals would be arrhythmic. But this shows that apparently the crygenes are essential for the circadian clock. If you don't have crygenes, you don't have a clock. And actually we have generated here the first let's say laboratory-made mouse without a circadian clock. And with two publications, very high ranking journals, we kind of entered this circadian clock field. And so we continue to work in that direction.
One, is the total amount of activity the same in the arrhythmic and the rhythmic mice?
Yeah, I mean, there are of course differences between between different mice. But if you have a mouse, if you would place this mouse, let's say either CRY1 or CRY2 single mutant mouse or a double mutant mouse, if you would place the animals in an LD cycle or in constant darkness, they have approximately the same amount of activity per 24 hours. There are of course inter-animal variation. Okay, then this is maybe the most difficult part of my presentation, but I would like to show it because it shows you how, in fact, I'm still, let's say, I get enthusiastic every moment again when I see how nature can kind of generate time. So basically what we have now, and this is a very simplified model because we found the cryptochrome 1 and 2 genes, other groups found clock and bemol, and there's also the period 1 and the period 2 genes. They are all involved in in the generation of time. But let's stick to the most simplified model. So we have here the cry gene. So this is the gene itself with its coding sequence required to produce the cry protein. In front of this gene you have a specific piece of DNA that acts, we call it a promoter, but it's in fact a piece of DNA that allows the gene to be switched on and off. So in this setting the gene is off. If you now allow the clock and bemol protein to bind to this what we call e-box, the gene is switched on. So this means that an RNA copy is made of this gene. So this gene is on the DNA. The DNA cannot leave the nucleus. So we need to make a RNA copy of the gene that goes out into the cytoplasm. There this RNA is used to synthesize protein. These proteins go back into the nucleus and in the end accumulate in the nucleus. So that's what I've indicated over here. So you see the cry protein being produced. What happens then is that the cry protein can bind to clock and bemol. So if the concentration of cry protein in the nucleus is high enough, it will bind to clock and bemol. And look what happens now. So clock and bemol are degraded and in fact the gene is switched off. So basically there's no longer RNA produced. As you can see here, there's no longer protein produced, so protein levels go down again. And after a while you're back to where you came from, back to the start. So basically you can allow this cycle to start all over again. And this is a way how nature, how molecules and DNA and protein can generate an oscillation. A clockwork. And every time I think about it, I get impressed about it. And so basically what we are talking about is what we call negative transcription translation feedback loop. So you have a gene and the protein that is synthesized from this gene switches down its own synthesis. So molecular oscillator. And of course we biologists would like to visualize everything. And what we have done here is that we have introduced what we call reporter gene into this cell and in the mouse. So this reporter gene contains what we call this e-box sequence that allows a gene to be switched on and off by clock and bemol. But instead of having a functional cry or whatever gene, we have placed the luciferase gene from the firefly behind this promoter. So basically when this clock is ticking in this cell, also this luciferase gene will be temporarily switched on and off. And if you now give the right substrate to this cell, so the substrate that is required to generate light, then you see that this cell is also producing light in a rhythmic manner. So basically, you see here an individual, let's say Rolex, the Rolex of an individual cell. You see the clock ticking.
Why does it happen in every cell?
I mean, it depends. You can take a cell. You can take a dish with cultured cells. You can transfect. It's a difficult word. You can introduce a clock reporter into these cells. And basically, if you do an experiment in cultured cells, maybe 1 in 100 cells will pick up this reporter. But you can also make a transgenic mouse. So you can bring this reporter gene into the fertilized oocyte of a mouse, and you can place back this. Then this reporter gene will be integrated in the DNA. Then you bring back this oocyte into a pseudo-pregnant female, and then you allow this fertilized egg to be to develop into a mouse, and you get a transgenic mouse that has this reporter in every cell. And that is it's a good point if you make it right here, because it brings you to the next slide. Making use of those mice with these reporters, clock reporters in every cell, it has been shown that we do not only have a central clock in the SCN, a central clock that is receiving light information to be reset daily and to be synchronized with the light-dark cycle, but basically we know now that every cell in our body does have a circadian clock. So here you see the kidney and the liver. All those kidney and liver cells do have a clock. In fact, all the cells in our body, even our white blood cells, our bone cells, whatever, they do have a circadian clock. There's only one difference, that those peripheral clocks, those peripheral cells, those clocks, they're not sensitive to light. Even if you would have a skin cell that is quite on the surface of our body, it does not respond to light. Where it does respond to is, in fact, stimuli that are coming from the SCN. So the SCN is releasing in a rhythmic manner compounds, peptide hormones that in the end synchronize the clocks in the periphery. So also those clocks are kept in a 24-hour rhythm by these compounds coming from the SCN.
If there would have been a mutation, if there would have been a mutation within the cells and it would not break itself down, would it then, you then always sleep or always be awake?
That's a good point. I think, so if you don't have a clock, there's no rhythm anymore, as I've also shown you those mice. So you will be, so you will sleep, let's say, for a short period and be active for a short period, so there's no rhythm anymore. It's not that you get stuck in one, let's say, position. Although there are situations, it doesn't come back in my presentation, it may go too far to discuss it now, but, so we have this cry-1, cry-2 knockout mice. Let me actually briefly mention it. So we have this clock this model over here. So if you have a mouse that doesn't have any cry gene anymore, if this is the case, then all those EBOX genes are continuously switched on, because clock and BMOL activate this type of EBOX genes, but there's no cry, so it will not be shut down. The opposite situation will occur if you have, for instance, a clock or a BMOL knockout mouse. If you have a mouse that doesn't produce BMOL anymore, then those genes will never be switched on. So you can have, I will come back on EBOX genes in a moment, but you can have a situation where certain genes are continuously on or continuously off. So there might be processes that are, depending on the type of mutation, are continuously on or off. But for the sleep-wake cycle, so if you lose your rhythm, you will not be, let's say, awake for always, or you will be asleep for always. That's not the case. Okay, so here we've shown that, in fact, every cell in our body. Oh, sorry.
Yes, the question is still there. Because of the eye in your picture, I was wondering how the blind are managing.
Yeah, good question. There are two situations. So if you would have a mouse that does not produce, that does not generate any eye because of a genetic defect in the development of the eye, you would have a mouse that would continuously follow its own internal clockwork. So irrespective on whether you have a light-dark cycle or not, so they will follow their own internal clock. However, if you would have, and the same holds for humans, if you would have a person that, for instance, due to a traffic accident would lose both eyes, would have two glass eyes, those people will not receive, so in those people the SCN will not receive light information anymore, so those people would, what we call free run, they would live according to their own circadian clock. However, if you would have a person that gets blind because it's losing its, he or she is losing its photoreceptors in the retina, so there's a disorder called, or a condition known as retinal ablation, which means that your rod and cone layer will become detached from the, let's say, the eyeball. Those people get visually blind, but they are not circadian blind because at another position in the eye there are still what we call ganglion cells, kind of connecting cells that also do have a photoreceptor protein. It's called melanopsin. We only know that for a couple of, let's say, for 5-6 years now. So those photoreceptor cells are still present, so then the eye still gets light information. So if you lose your eyes, you're circadianly blind. If you lose your rods and cones, you still see the light in terms of clock resetting. Now, even if you would have so you could see now this circadian system as a kind of, call it symphonic orchestra, where the SCN acts as the conductor. And this conductor gives the signals to the various sections in the orchestra, the violins, the contrabasses, the horn section. And so this director takes care that all these sections collaborate nicely together, and in the end give one harmonic piece of music. But the SCN is doing the same. It is organizing, or let's say it's conducting the clocks in this in the peripheral tissues, and in fact this whole circadian system now kind of provides a temporal organization of the body. So it takes care that all the organs are call it active on the right moment of the day and also in the right order. For instance, before you start to eat, the stomach already starts to produce digestives and your intestines, etc. And then at a later moment, your liver starts to become active in order to take out the toxic compounds from your food. So everything is working nicely together now. And this slide shows you that even culture itself, as we already discussed, even culture itself do have a circadian clock. There's only one but, and it is, and you can explain, you can understand why. I just told you that those peripheral cells get time information from the SCN. So once a day they get a stimulus that synchronizes those clocks in the liver and in the kidney. If you now culture your cells outside the body, or if you take a liver slice and bring it in culture, you will see that initially those clocks will run synchronously. But you can imagine as those cells no longer receive daily stimuli from the brain, what will happen in the end is all those clocks start to tick asynchronously. And time flies. Then it's no use of having a clock if this clock is not connected to output processes in behavior, physiology, and metabolism. Okay, here I show you an example. So we already mentioned, I already gave a kind of hint that there may be more EBOX genes. So in order to link this clock to these processes, there's a whole bunch of what we call EBOX genes, I call them 1 till 4 over here. And you can see that also those genes under control of clock and all are oscillating, so you get rhythmic expression of these proteins. So initially we thought, okay, there's only one series of EBOX genes. We now know that among those genes there may be other factors that in turn allow other genes to be expressed in a rhythmic manner. And this rhythm not necessarily needs to be in the same phase as the phase of this EBOX gene. This is shown over here. So you can also have other proteins that are oscillating in another phase. And actually what we know now by a technique that we know as microarray analysis, it's a technique in which you can analyze the activity of all 25,000 genes in one and the same sample. So if you take a mouse and you would take a liver sample every 2 hours and measure the activity of all genes, you would see that a surprisingly high number of genes is oscillating. So initially we thought, okay, maybe it's a handful of genes that is connecting this clock to, let's say, this output process, but it turns out that it's 5 to 10%. And this we know now for let's say some 8-9 years. And this is striking because it means now, you see here this classical chart of this Bergen chart with all the biological processes going on in our cells and in our body. If 5 to 10% of our genes is under circadian control, it also means that out of all these processes, there's hardly any process that escapes from being under circadian control. So you have this cascade. For instance, if you degrade your glucose molecule and convert it into energy, it's a cascade of, let's say, 20 proteins. So this requires 20 genes. 5% of 20 is 1. So at least one of those genes is expected to be under control by this clock. So the whole process is under control by this clock. And now you can say, who cares? People on average hardly care about the circadian clock. But I think we should. And so time matters, but is largely ignored in science. And for instance, if you look at in vivo experiments, in vivo means that you do the experiment with animals, live animals. Many people use rats and mice. These are nocturnal animals. However, most people, at least in the past, it's changing nowadays. We are scientists, we are humans, we live during the day. So we do our experiment during the day. We take the mice, the animal from its cage. We do an experiment here. This is, for instance, a grip strength test. So you look at the force that an animal can develop in its forepaws. People get the animal, they take the animal from their cage during the day. So when the animal is actually sleeping, and I would ask this, you can ask the same researcher this question. So what would happen if we would wake up Epke Zonderland in the middle of the night, ask him to perform his trick at the bar? Would he have obtained gold? Probably not. So I think you should simply do this kind of experiment during the day of the mouse, which is our night. And this not only holds this behavioral study, also when you look at the time of the day where people collect blood or tissues from a mouse or from an animal, or they expose the animal to drugs, they simply do it at the wrong moment of the day. So what do your data tell you? And I will show you an example in a moment. The other thing is that, so the same holds for experiments with cultured cells. How many of the researchers do know that their cells still do have a circadian clock, and that this clock is ticking asynchronously when they have the cells in culture? And how many people would realize that even a small shift in temperature could temporarily reset those clocks? So you should take this into account when you do your experiment. And also in medicine. So I think that many disorders, so that the circadian clock is playing a role in the etiology, so the mechanism behind the onset, the appearance of disorders, as well as it's playing a role in the treatment and the prevention of disorders. And I give you some examples in the remainder of my presentation. So for instance, the impact of the circadian clock, sleep and mood disorders. You will all recognize the fact that you have people in your environment that are kind of morning people that perform best in the morning, other people perform best in the evening, so the larks and the owls. And you can ask people to fill in a questionnaire, so if you fill in this questionnaire will tell you what kind of, what we call chronotype you have. So what kind of person are you, an early type, normal, what is normal, but a normal type, or a late chronotype. So this is the distribution of chronotypes over the population. Now you can explain this now. So for instance, if you are morning people, so I plotted here a kind of called imaginary gene. So this is oscillating. Now the idea is that people, and it has been shown that people with an early chronotype do have a fast ticking clock, while people with a late chronotype do have a slow ticking clock. And you can immediately see the impact over here. So you see that this clock is ticking. So this is the moment of resetting. So the clock is ticking. Genes are oscillating. If you have a slow ticking clock, you have a call it long wavelength. There's a fast ticking. You have a short period. And what you see now, suppose that for this imaginary gene, this is the level where you start to fall asleep. So if you have a normal chronotype, you will fall asleep, say around 12 o'clock in the night. If you are a late type, you may fall asleep later in the night, 1 o'clock. If you have an early chronotype and a fast ticking clock, you may fall asleep at 11 o'clock. So this explains the chronotype and its impact on, in this case, the sleep-wake cycle, which you can translate this phenomena to all kind of other parameters in physiology, behavior, and metabolism. Now, the other thing to know is that the chronotype is not constant, it's changing over age. So if you're a young person, you on average are an early person, early bird. When you become a teenager and grow into adolescence, you see that this chronotype is moving into that of a late person, a late chronotype. And when we start to age again, we get early birds again. And when you look at the homes of the elderly, you see that on average, most elderly, most older people are morning type of people. So they wake up very early during the day, and they fall asleep pretty early in the evening again. And on top of that, they have many naps over the day. That is also because their circadian clock is disturbed. The amplitude is getting weaker. I don't have time to get into detail there, but upon aging, also the amplitude of the circadian oscillator is reduced. So you get a weaker clock. So you could see this as a kind of gliding scale. The larks and the owls at the two ultimate sides, but you can even go beyond this. So you can have a very extreme early or late phenotype, and when you have this, you may even suffer from it. So this is called. So these are two circadian disorders. Advanced and delayed sleep phase syndrome, in which patients fall asleep either very early in the evening to wake up in the middle of the night and after they can't sleep anymore, or you can have a late phenotype. People fall asleep, let's say 3 to 6 o'clock in the night, so early in the morning, and then they wake up next day noon. These are inherited disorders, so there's certainly a genetic component over here, and the problem is that those people are forced by their bosses, by their schools, to live, let's say, outside the, they're forced to live out of phase with their internal circadian clock. And this gives rise to a chronic sleep debt, that they don't sleep enough, they're chronically tired, they perform less well, and they even, to a large extent, will develop depression. So this is very, let's say, invading. Okay, now how to determine whether someone has a clock defect or not? So what you can do is you can do this type of this bunker type of experiment where you house people in a time-free room, and you simply look how their behavior is under free running conditions. So if you don't give them light information, house them under dim red light, you see that those people also follow their own clock. This is an example of an advanced sleep phase syndrome patient. So we've just seen that the human circadian clock has a periodicity of 25 hours. You see that this patient has a clock that runs way faster. So the main question now is in the field is which clock genes are affected, how can we diagnose it, and can we in the end develop a treatment to allow those clocks to perform normally. Now one of the things that we did is we developed a method, but not only we, many other labs did as well, where we could take a skin biopsy from a patient or a normal subject. We can bring the cells in culture, then as Alan just asked, okay, so we can introduce this clock marker into those cultured cells, and then we can follow those dishes. We can place those dishes in an incubator, and in this case it's a special incubator that contains a couple of photo multiplier tubes that can measure the amount of light that is coming from those cells. And what you can see here is that you can visualize the oscillation, you can visualize the clock of those cultured cells. And what we're doing now is that we are using this assay to determine whether patients advanced, so to identify patients with advanced and delayed sleep phase syndrome, just on the basis of the periodicity of the circadian clock. Once we identify those patients, we would like to see which genes, which clock genes or which other unknown genes are defective in those patients. I also would like to mention one slide, seasonal affective disorder. So seasonal affective disorder is a disorder in which patients get depressive in autumn and in winter. And in spring, they get rid of those symptoms again. So it's a temporary, let's say, disease. It is believed to relate to the amount of light that is perceived by the eyes during the day. And when the days get shorter, there's a chance that people don't get enough light to keep their clocks riding and this can give problems. It's also shown that bright light therapies in some occasions can cure people from this depressive, let's say, moods. And indicative for the fact that it's highly related to, let's say, day length is the fact that you see this disorder and a high incidence at the, let's say, northern hemisphere. For instance, in the northern part of Finland, up to 10% of the population is suffering from seasonal affective disorder. And it's also nice to think about where this seasonal affective disorders may come from. So a couple of times, years ago, it was speculated that maybe seasonal affective disorder was a kind of remnant of, let's say, hibernation, so the winter sleep. Animals at the very northern part of the world, living close to the poles or in the northern parts of Finland and Sweden, whatever, you see that they start to sleep in the winter. So when the environmental conditions become less favorable, rather than trying to survive, they prefer to kind of slow down all the body processes and sleep for a couple of months till the environment is favorable again. And since many people encounter in autumn this feeling, so they don't like to do this or that, they're not really depressive, but they also they're not as, let's say, they don't have as much energy as during the summer and spring. So that's why I believe that perhaps this let's say this seasonal affective disorder is a kind of remnant of hibernation, so slowing down of body processes. I'll come back on that at the end of my presentation. So another example of time matters, but it's largely ignored is the impact of the circadian clock, for instance, on the prevention of cancer. I just mentioned to you that we have our DNA with over 25,000 genes, And I already mentioned that this DNA can be damaged by ultraviolet light. That's not the only thing. There's also many compounds in our environment that can damage this DNA. It can be cigarette smoke, can be this exhaust gases, industry, solvents, whatever, your burnt barbecue sauces. These can damage the DNA, and this is exemplified right here. So look at this sequence, AACT. What can happen now that, for instance, this T is modified, and this modification, cells don't like that. So this can cause a cell to die. And in fact, if this happens, you see a kind of toxic response. The cells die. But it can even be worse. Maybe the cell doesn't die, but still continue to duplicate its DNA. And what can happen then is that opposite this T, where you always find an A, in a newly synthesized DNA, there can be a G. So in the end, you get a change, a base pair change. And we call this a mutation. And this mutation, in fact, can, for instance, activate oncogenes, so genes that are required that make you more vulnerable to getting tumors, or it may suppress genes that normally suppress tumor growth. So you may get cancer. And so this is an experiment that shows that exposure to DNA damaging agents, that time matters. This is a classical toxicological experiment where people take a mouse, they expose it to cyclophosphamide. In this case, we did the experiment ourselves, and the purpose will be clear to you in a moment. We expose the animals to cyclophosphamide, which in the end gives DNA damage. It cross-links the DNA and causes cell death, so it's a toxic compound. But in order to become toxic, it first needs to be activated into an active form in the liver. Now, so what we would like to know now is, okay, what happens, how does the liver of this mouse respond to exposure to cyclophosphamide, and which genes are switched on or off, which genes are activated, become more active or become less active. And you can do that by this technique that I already mentioned. So you can do a microarray, a chip experiment where you look at the activity of all the genes in a cell or in the liver, and by identifying how those genes respond, you get insight in the biological processes that respond to exposure of the cells of the liver to cyclophosphamide. And this gives you insight in the toxic response. Now here comes the experiment. We are lazy scientists. We do the experiment during the day because then we are in the lab anyway. But we take the animal from its cage, the animal is sleeping, we inject it with cyclophosphamide at 2 o'clock in the afternoon, at 6 o'clock in the afternoon. We kill the animal and we isolate the liver, and then we look at the genes that are switched on or off after exposure to cyclophosphamide. Then you see in this graph that there are some 2,000 genes that are becoming more or less active upon exposure to cyclophosphamide. But what happens if we would have exposed the animal under more physiological conditions, so that is during our night, so during the day of this animal? And that's what we did as well. So here we exposed the animal to cyclophosphamide at 2 o'clock in the night, and we isolated the liver at 6 o'clock, and then you see that there's a huge difference in the amount of genes that is becoming more active or that is inhibited by exposure to cyclophosphamide. And you see that it's only a very small number of genes that is overlapping, so it's about 50 genes that are switched on independent of the moment of the day, or switched off independent of the moment of the day. But there's some close to 2,000 genes that are only responding during the day. And we, of course, we didn't do this experiment without an underlying idea. So what was known from the literature is that if you give the mouse cyclophosphamide at 2 o'clock in the afternoon, those animals pretty well tolerate the compound, depending on the dose, of course. But there is a dose at which those animals don't suffer that much from being exposed to cyclophosphamide. While if animals are exposed to cyclophosphamide, same dose at 2 o'clock in the night, they die. So apparently it's a matter of life and death. And now comes the whole point. Apparently in this set of 2,000 genes that are specifically activated or inactivated during the day, there must be genes that somehow protect these animals from being killed by cyclophosphamide. Now, this is a clear example of what we call chronotoxicity. So here, molecular biologists, chronobiologists work together now with toxicologists to further unravel the underlying mechanisms of toxicity. And in fact, sorry?
5 minuten afgerond.
5 minuten afgerond. Oké, laat ik dan misschien de meest. Oké, misschien wel. Even kijken, dus we hebben hier nu.
In English. In English for the recording.
English.
5 minuten, oké, nou dan hebben we so our industry is producing chemical compounds in tons and tons during the year. They produce new types of compounds that can be toxic, can damage our DNA, be genotoxic, or can be carcinogenic. Our society wants us to test all these compounds for toxicity, etc., etc. And the other thing that today this is hardly achieved without the use of laboratory animals. But at the same time, our society also wants to get rid of those animal experiments. So basically, what we are doing now is that we are trying to use this genomics approach in combination with cell and tissue cultures to see if we can develop what we call risk profiles for classes of chemicals. So if we have a chemical that when given to cultured cells gives a certain response, certain genes go up, certain genes go down, this can be characteristic for a compound that is, for instance, carcinogenic for the liver or whatever. So we hope to produce these kind of risk profiles so that we can get rid of those animal experiments. And what we are doing now is that we are considering the factor time in these assays. So basically what we are doing is that we, so you can take cell cultures and you don't consider time, you simply do the experiment in a population of unsynchronized cells. Or what we are doing, and what is novel in this field, is that you can first synchronize your cells with a compound and then expose the cells at defined moments of the circadian day. As you can see here, you can get a way larger response, and you can also get information on whether a compound is more or less toxic during a certain moment of the day. And why would we like to do that? It is because this information can be used in our society, and that's where we get into contact with, for instance, doctors or people from the ARBO departments. So you can use this information to improve the working conditions for laborers. If you find a certain compound that important that you have to work with it in industry, and for instance you need to replace a vessel once a day, then you can tell your people, okay, you have to protect yourself in order to avoid that you are exposed to this compound. But you can always have a situation where a glove may be ruptured. So if you know that a compound is more toxic to the human body in the evening than it is in the morning, you can advise people to work with this compound in the morning rather than during the day. So you can improve what we call labor hygiene. And recently the industry, but also this ARBO medics, they got very much interest in this type of work. And you can go even one step further, and it is that there are many toxic compounds that are used as medicine. For instance, if you want to kill a cancer cell, you can simply use a cell-killing compound, and many of those drugs are used in as chemotherapeutic agents. And the point is that, but I don't have time to get into detail there, is that even in therapy you can give drugs at the right time of the day, and it will help you to improve, let's say, the efficacy of the drugs. In a nutshell, cancer cells are. Sorry, let me ask you. I'm running out of time. You have to believe me that by giving drugs at the right time of the day, they can be more or less effective. And in the case of cancer drugs, you can have more or less side effects. So you should give drugs to a patient when it is best tolerated by the body. And this is what is happening already in France, and where we are really making a start with now in the Netherlands. And there's so much more to come.
Thank you.
Thank you for